Bismillahirrahmanirrahim. Thank you, Dr. Zahid. First of all, I want to thank organizing committee for this nice event. Yesterday, Dr. Hamad also present serum-induced tibial dyschondroplasia, and he get excellent result of plastum extract against this disease. Today, I will discuss about chondroitin sulfate reverse tibial dyschondroplasia via CHST11 beta-catenin pathway. So this disease is actually a tibial dyschondroplasia. It affects chicken and a severe welfare issue. Actually, it is caused by serum, which is normally used in grain storage. So due to contamination, it affects the chickens, and affected chicken may show some kind of sign like lameness, spray out leg posture. They are unable to take the food. That's why their body weight is decreased due to inability to take water and food. First of all, we discuss its background. So this disease is very important in case of economic point of view. It can approximately losses 30% issue and prevalent in China. So due to during the processing, when meat is processed, it affects the meat quality. That's why they focus on this disease. In this trial, we get excellent result of glycosaminoglycan, which produce chondroitin sulfate. So chondroitin sulfate is actually chondroitin sulfate actually restore the growth rate cartilage. When it is restored, so the growth is increased, and chicken can go towards the water and take normal food. The drug trial of our it include 21 days. So when we give feed to normal chicken for 1 to 21 days, in control group we give normal food, while in case of TD group we give serum from 4 to 7 days. When serum was given, so animal get the tibial dyschondroplasia in this disease. So this disease is induced, and in case of TDLC and TDSC, we give chondroitin sulfate. Chondroitin sulfate is induced along is given along with the serum. Chondroitin sulfate is given along with the serum. So chondroitin sulfate is give protective effect. At the 21 days, all the chicken were normally slaughtered, and we get the tibia bone and other important organs for further processing. Along with in vivo trial, we did also some in vitro experiment for the confirmation of this effect of chondroitin. So in case of in vitro trial, we did some knockdown effect. We get some beneficial effect by knockdown of CST11 gene. This gene is normally involved with beta-catenin pathway. When this gene is knocked down, so disease severity is increased. So in this way, we decided that this pathway is very important for the pathogenesis and the occurrence of this disease. In case of in vitro trial, we get the chondrocytes from 3-day-old chicken and did the culturing along with the serum in case of TD group, while in second group we we knocked down the gene of CST11. In third group, we overexpress this overexpress this gene. In last group, fourth one, we had chondroitin in along with knockdown gene. Here you can see the results of in vivo trial. You can see in case of TD chicken, animally birds were lying on the ground. They were unable to move. While after adding CS, they get normal posture. The birds were moving. They getting the feed normally. After slaughtering, you can check. Here we can see the results. After slaughtering on 21 days, there is again problem of growth plate. Growth plate is enlarged. There was white opaque mass you can see here. But after the administration of CS in low concentration and high concentration 800 and 1600, you can see there were improvement in the growth plate and their growth plate size is decreased and cell proliferation and differentiation started. So we can say CS effectively mitigate lameness and extent of cartilage embolism in tibial dyschondroplasia. You can see the growth parameters. The growth parameter include tibia mass along with tibial length is decreased in case of tibial dyschondroplasia, while other parameters like tibia index is increased in TD chicken. While the CS administration normalize these parameters, chicken recover and their growth along with the bone development is stabilized near to the normal group. So if you see the body weight at 7, 14 and 21 days, there was significant difference in body weight gain of TD chicken and other group. So we can say CS improved the growth performance in the chicken. This is again in vivo results of H and E. You can see here in hypertrophic zone, there is vascularization properly in normal group, while in case of TD group, the vascularization was improper, cell were randomly, they cannot differentiate like normal in different zones. While after the administration of CS, the cell were differentiate in different proliferated and hypertrophic zone, and the size of growth plate is reduced, and the chicken started to recover and their bone formation started. So we can say CS restore normal secretion of matrix in resting zone, proliferative and differentiation of the cell, especially chondrocytes in proliferative and hypertrophy zone. RT-qPCR result indicate the expression of our important gene CST11. It is decreased, which is mainly important in this pathway, while the other related genes of this pathway are also showing less expression compared to normal groups. While the protein expression, including beta-catenin and other, also indicate there was down regulation of mostly protein expression. So growth performance related parameter genes and protein expression is affected in tibial dyschondroplasia chicken. While administering CS, there was some normalization of expression. In vivo trial indicate that cell survival rate on various concentration of CS, while immunofluorescent result indicate the cell shape and different expressions of proteins. In case of CST11 knockdown gene, here you can say the expression is significantly decreased. While in case of overexpression group, the expression of this gene of interest is increased. This is again RT-qPCR and Western blotting techniques were applied in different cell culture groups. The knockdown group indicate the expression on other related genes in the pathway, Wnt and beta-catenin pathway. So you can say these pathways mainly involved in cell differentiation and expression along with bone growth. CS restored the expression of cartilage development related to related factor caused by CST11 inhibition. The regular mechanism of CS on these genes is related to increased expression of beta-catenin. The conclusion of this study, we can say tibial dyschondroplasia, which is caused by serum or metabolic disorder, it affects the expression of CST11 along with protein expression of beta-catenin. This gene expression suppress the protein expression, which ultimately inhibit the transcription process in the nucleus. When this process is inhibited, ultimately extracellular matrix cannot synthesize the cell normally and cell differentiation didn't start. So birds will be suffer from tibial dyschondroplasia. But when we administer chondroitin sulfate, CS, it regulate, normalize the expression of CST11. When the expression of this gene is normalized, it will affect or enhance the expression, promote the expression of beta-catenin protein, which will enhance the transcription processes, ultimate normal ECM biosynthesis and cell proliferation will start, which indicate cartilage development process. This project was funded by various funding agencies, Shizang, Nature Science Foundation, Nature Science Foundation of China, Science and Technology projects in Guangzhou, and specific university discipline construction project. This project is conducted in China, in South China Agriculture University, by the team led by Dr. Hui Zhang. He is working as associate professor in South China Agriculture University. Actually, he have to come here and present his work, but due to some unfortunate condition, he cannot come. So he sent me his best wishes for this conference. And this is the pictorial view. You can see this group. This actually work is the presentation of this work actually is the effort of this team. Thank you.